Hello everyone, welcome back to my channel. In this video lecture, we will cover a new chapter of biology, that is sexual reproduction in flowering plants. So, first of all, reproduction ensures continuity of species generations after generation as the older individual undergoes senescence and die. Then, pre-fertilization structural events. Several structural and hormonal changes leads to formation and development of floral primordium. Then the inflorescence is formed that bears floral buds and then flower. In flowers, male is the androecium and female is the gynoecium, which differentiate and develops, in which male and female gametes are produced. Stamen, microsporangium and pollen grains. Stamen consists of long and slender stalk called filament and generally bilobed anthers. Each lobe contains two theca and then they are called as diathecious. This is the structure of the microsporangia anther. It develops and forms pollen sacs which contain pollen grains. Microsporangium is generally surrounded by four layers: the epidermis, endothecium, middle layer and tapetum. Innermost layer is the tapetum which nourishes the developing pollen grain. Then sporangious tissue, it is compactly arranged homogeneous cells which are present at the center of each microsporangium when the anther is young. Then comes the microsporogenesis, the process of formation and differentiation of microspores from MMC yani microspore mother cell by reductional division is called as microsporogenesis. The cells of this sporangious tissue undergoes meiotic division to form microspore tetrads. Then as the anther mature and dehydrate, the microspore dissociate and develops into pollen grain. Pollen grain represent the male gametophyte. Pollen grains are of two-layered, exine and entine. Exine is made up of sporopollenin, that is the most resistant organic matter known. It can withstand high temperature, strong acid and alkali, and no enzyme can degrade sporopollenin, and this is the reason that why pollen grains fossils are still found. Entine is the thin and continuous layer made up of cellulose and pectin yani pectocellulose. Then third is the germ pore, that is the aperture on exine where the sporopollenin is absent and it forms the pollen tube. Then a plasma membrane surrounds cytoplasm of the pollen grain. A mature pollen grain consists of two cells with nucleus, yani that is vegetative and generative cell. Vegetative cell is the bigger cell, contains abundant food reserves, have large irregular nucleus and is responsible for development of pollen grain. Then the generative cell is small and it involves in syngamy, yani it fuses with an egg and contains dense cytoplasm and nucleus. Pollen grains of many species, that is Parthenium, causes several allergies and bronchial diseases in some people and led to chronic respiratory disorders like asthma, bronchitis, etc. Viability of pollen grain varies with species to species and should land on stigma before this period to germinate. Pollen grains of large number of species are stored in liquid nitrogen at temperature 196 degree, that is called as pollen bank. Then the pistil, microsporangium and embryossac. Gynoecium may consist of single pistil, that is monocarpellary, or more than one pistil, polycarpellary, which may be fused, or free, that is if it is free then it is apocarpus and if it is fused it is syncarpus. Then each pistil has three parts: stigma, style and ovary. Megasporangium. Ovule is the small structure attached to placenta. Funicle, stalk by which ovule is attached to placenta. Helium, junction between ovule and funicle. Integuments, protective envelopes. Micropyle, small opening at the tip of ovules into where the pollen tube enters. Chalice, basal part of the ovule. Nucleus is the 2N, that is mass of cell enclosed in integuments, has abundant food reserves. Then microsporogenesis, it is a process of formation of megaspore from megaspore mother cell by meiotic division and this process takes place in ovule. Ovule differentiates a single spore megaspore mother cell in micropolar region of nucleus. Then MMC undergoes meiotic division that results into the production of 4 megaspore. Then one megaspore develops into, out of the tetrads, only one megaspore develops into female gametophyte and the rest 3 gets degenerated. 3 cells are grouped at micropylar end to constitute agapparatus, and 3 cells at chrysalis end to form antipodal cells, and at maturity embryo sac is 8 nucleated and 7 cell structure. Then pollination is the transfer of pollen grains from anther to stigma. Autogamy, transfer of pollen grain from anther to stigma of the same flower. Cleistogamous, flowers which do not open. Cleistogamous flowers are autogamous as there is no chance of cross pollen, and cleistogamous flower produces a short seed set even in the absence of pollinators. And chasmogamous, export anther and stigma. Then geitonogamy, transfer of pollen grains anther from stigma of different flower of same plant. Then geitonogamy is functionally cross pollination involving a pollinating agent. Genetically it is similar to autogamy since the pollen grains come from the same plant. Then xenogamy, transfer of pollen grain from anther to stigma of different plants, flower of the same species. Then wind pollination, the pollen grains must be light, non-sticky and winged. The anther is well exposed, stigma is large and feathery, flower is one ovule arranged as inflorescence. And then water pollination, pollen grain is produced by mucilaginous covering. Then examples of fresh water, that is Vallisneria and Hydrophila. Main features of wind and water pollinated plants are produce pollen grains in large number and do not produce nectar. Insect pollination, the flowers are large, colorful, fragrant, rich in nectar. Pollen grains are sticky and stigma is too sticky. They give rewards to the pollinators too, like nectar and edible pollen grains as food, and provide safe place for laying eggs like Amorphophallus and Yucca. And outbreeding devices, the various mechanism discourage to self-pollination and encourage cross-pollination, as continued self-pollination leads to inbreeding depression. It includes pollen release and stigma receptivity not synchronized. Then anther and stigma are placed at different position. Then inhibiting pollen germination in pistil. Then production of unisexual flowers. Then pollen pistil interaction. The pistil has the ability to recognize the compatible pollen to initiate post-pollination events that leads to fertilization. Then pollen grains produce pollen tube through germ pore to facilitate the transfer of male gamete to embryo sac. Then artificial hybridization, crossing different varieties of species, hybrid individual with desirable characters of parent plants. Then emasculation, removal of anther, bagging, flower covered with bag made up of butter, prevent contamination of stigma from unwanted pollen. When bagged flower attains receptivity, mature pollen grains dusted on stigma, rebagged flowers, fruits allowed to develop. Then double fertilization. After entering the one of the synergids, each pollen grain releases 2 male gametes. One male gamete fused with egg and other male gamete fused with 2 polar nuclei, that is called as triple fusion. And to produce triploid primary endosperm nucleus, since 2 types of fusion takes place in an embryo sac, the phenomenon is called double fertilization. Then the endosperm, the primary endosperm cell divides in many times to form triploid endosperm tissues having reserved food materials. In the type of endosperm development, free nuclear type, cellular type. A non-albuminous, then albuminous. In non-albuminous, endosperm completely utilized before maturation, and albuminous, a portion of endosperm remain in mature seed. Embryo develops at the micropolar end of the embryo sac where the zygote is located. Embryogeny, early stages of embryo development. Embryo consists of embryonal axis, cotyledon, spline and radicle. Monocotyledon seeds contain scutellum, cotyledon and chlorhiza. Seed is a fertilized and mature ovule developed into seed. It consists of cotyledons, embryonal axis, seed coat, double layered, formed by integuments, that is testa and tegmen. Micropyle, small opening on the seed coat, facilitate entry of water and oxygen into seeds for germination. Helium, scar on the seed coat, seed, albuminous, non-albuminous, perisperm, remnants of nucleus that is persistent, then dormancy, state of inactivity. The wall of ovary develops into wall of fruit called pericarp, and in true fruits only ovary contributes in fruit formation by, and false fruit thalamus also contributes in fruit formation. Apoximosis is the form of asexual reproduction that mimics sexual reproduction in which seed is formed without fertilization. It is the formation of amocytic seeds, diploid cells developed into embryo without fertilization. Cells of nucleus surrounding embryo sac protrudes into the embryo sac, developing into the embryos. Polyembryony, occurrence of more than one embryo in a seed, and often associates with apoximosis. So this much for today's video. Hope you liked it and understood it. Thank you for watching my video. Till then, bye bye.